Jacob Kiner for the Unit 3 discussion video. It's understood that DNA, for the most part, encodes the building blocks for life, being proteins. Most of DNA is coded to help produce proteins that form the various parts of us and pretty much any other living thing, from the smallest cell to us. However, it's understood that a lot of DNA is known as non-coding or sometimes even as junk DNA because they don't code for specific proteins. There's only a certain amount of proteins out there, but there's a vast majority of DNA sequences that don't necessarily code for a single protein. So for some time, this has been considered a mystery, like why do we have DNA that does not code for specific proteins? Sometimes they code for RNA, sure, but a lot of stuff doesn't seem to have a specific function that we didn't know of at that point. However, in this article, Bernaldi's article here, he starts to connect some of the dots that may give better insight into what some of this quote-unquote junk or selfish DNA might actually do. So for one thing, it starts breaking down two different quote-unquote superfamilies of isochores. GC, guanine, thymine, poor, and GC rich isochores. The GC poor isochores are correlated with lamina associated domains, or LADs, LADs as I might be calling them, and the GC rich coordinate with TADs, or topologically associating domains. And both of these can be identified through mapping or through the replication timings that can be studied. So these two classes can also be distinguished by their topology, specifically by looking at the oligo A's and the oligo G frequencies and the heights and depths of the peaks and valleys in these frequencies. Oligo A sequences are found in LADs or LADs, and they kind of form a more rigid or stiffness in bending and some slight bending, but overall just more of a rigid structure, as opposed to the more higher frequency in oligo Gs, which allow for, among other things, are found more frequently in TADs, and they form more into loops. These things are found with help in the structures of the chromatin. It's also noted that the chromatin that are carrying with the corresponding, they have a higher number of corresponding GC poor isochores, have actually shown a more distant intrachromosomal interactions, while GC rich isochores show more localized interchromosomal interactions. So all these understanding coming from these quote-unquote non-coding DNA, and these non-coding DNA actually help with the structure and formation of these, which help differentiate between different things. We've noted different concentrations of GC rich and GC poor in even as big as differences as warm-blooded or cold-blooded animals in the animal kingdom, among other things. So this non-coding, while it doesn't create specific proteins per se, actually helps define a lot of different traits within DNA. But what does this all mean? According to Bernaldi, the better understanding of the various properties of these isochores based on their structures and the understanding of the chromatin's physical structures there help with a better understanding of what this DNA does and insight to why so many of these actually do play a part. My personal opinion on the article, not gonna lie, was a little tough to understand, but I think it does do a good job of connecting some dots that allow for a less dismissive argument of just that the DNA is just useless or selfish, even as if they have an agenda. And this allows for a better understanding of this DNA and why it plays such an important part in our genome, especially as the human genome has more or less been fully sequenced, and allows for better understanding of why we are what we are and how we do what we do in a very micro biological sense.